So it's going to be a little bit weird recording-wise, I think, just because my screen isn't registering touch on the big screen. But I think I've got it figured out. Think being the operative word here. Haha. So we are talking about alternative methods for carbon fixation. Ooh, that's gigantic. That's fun. Okay. Alternative methods for carbon fixation. So the way photosynthesis works, or the way the Calvin cycle more specifically works, is that Rubisco takes carbon dioxide out of the air and fixes it to our into our cycle. But life isn't always perfect. There are a lot of different factors that affect photosynthesis, especially in hot, arid climates. So, alternatives to carbon fixation. Have evolved in hot, arid climates. So we will talk a lot about the evolutionary connections, especially in this lesson. Yes, sorry, my brain's a little frazzled. Getting this to work. So, in really, really hot, really, really arid climates, deserts, all that fun stuff, plants have adapted to be able to carry out photosynthesis in environments where water is not really available. So they can't carry out what they normally do. So plant leaves have stomata, our openings where our CO2 comes in and our oxygen comes out, right? That is also the site of transpiration where like water evaporates directly from plant leaves. It's not super great in really, really hot, really, really arid climates because if we have those stomata open, it can suck all of the water out of our plant, and that's not super great. So we're going to start with a little evolution connection. So we have to balance photosynthesis and water loss. We have to balance photosynthesis and water loss. We have to figure out a way for plants to still maintain their levels of photosynthesis that help them stay alive without losing so much water that they can't carry out photosynthesis anymore. So like I said, our stomata are a little opening in plant or in leaves or in plants that let CO2 in and oxygen out. There has to be a way for carbon dioxide to get in and for oxygen out. Plants don't have lungs to help carry that stuff in and out. They have these. So just as our mouths and our noses is how our waste gases get out of our body and the good gases get into our body, plant leaves have stomata. This is, like I said, the site of transpiration. Transpiration is evaporation from plant leaves. It's one of those steps in the water cycle that some people hit on and some people don't. It's just a fancy word for evaporation, but out of plant leaves. Let's see. When we have low levels of CO2, when carbon dioxide is sparse and oxygen is high, that's not super great for our plant cells. Conditions will favor a process called photorespiration. Conditions favor a wasteful process called photorespiration. This is kind of where we get into our evolution thing. So, whenever our stomata aren't functioning properly, we don't have enough carbon dioxide being let into the cell. We have too much oxygen building up. That's not super great for what a plant cell needs. It will trigger this process called photorespiration. Photorespiration uses light, consumes oxygen, and releases CO2. So, photorespiration uses light, consumes oxygen, and releases CO2. So if we've got too much oxygen and not enough carbon dioxide, the cell will revert to this process right here. This uses ATP. Not super great. It decreases photosynthetic output. So even though it is releasing our inputs to photosynthesis, it's not increasing our photosynthetic output. It is costly and pointless. That was a weird growl sound. So we're gonna run through an example really quickly. So most plants, or plants that we call C3 plants, their first organic product is 3-phosphoglycerate. Three-phosphoglycerate is a 3-carbon sugar, hence C3. So most of our plants, our flowers or trees, etc., are C3 plants. And on super, super hot days, our stomata will partially close. It's really, really hot outside. Our plants don't want to lose the water that they have because that really, really hot day will kind of evaporate that out of them. So our stomata will partially close. When the stomata partially close, there is a decrease in CO2 because we can't let it in. And that starves the Calvin cycle. So our light reactions will go as far as they can with the NADP plus and ADP that they have. Eventually, they're going to use all that up, and we're just going to have our store of NADPH and our other high electron carriers. And what will happen is Rubisco will add, that's not a word. Rubisco will add oxygen, not carbon dioxide, to the Calvin cycle. Rubisco's just doing its job. It's just trying to create this compound. So Rubisco, if there's not enough carbon dioxide, we'll just add that oxygen. And when it adds that oxygen to the Calvin cycle, our 2-carbon sugar, or 2-carbon compound, 2-carbon compound will leave the chloroplasts where peroxisomes and mitochondria will release CO2. That's photorespiration. We've got our products coming from the light reactions, our NADPH and our ATP. They're going to go to the Calvin cycle just like normal, but instead of having our normal kind of Calvin cycle process where Rubisco will fix CO2 to our 5-carbon sugar, it will fix oxygen. So instead of having a 3-carbon product, we will have a 2-carbon product. That 2-carbon product will get sent to the mitochondria or the peroxisomes, and that will release CO2. Because remember, like I said, photorespiration uses light from the light reactions, will consume oxygen because it's going to add it to the Calvin cycle and release CO2, which are released by the mitochondria or the peroxisomes. This seems very, very counterintuitive. Why photorespiration? Why do we have this process if it's not doing anything productive for the plants? We don't know. We can't tell for sure. We think maybe it's an evolutionary leftover. A kind of, I guess leftover is the best word for it, leftover use of Rubisco. Because Rubisco is such a common enzyme in cells, we think it's one of the most common proteins on Earth. I think it's been around a while. So eventually, so like way back, we think it maybe had another kind of use or another process, and then it found its kind of home in photosynthesis, but it still maintained the ability to take oxygen and put it into something that would look like the Calvin cycle way back. We also think that it could maybe provide protection. Maybe that much oxygen in a plant cell could be considered toxic. So it's worth it to use energy to get rid of that oxygen. That could be another example. But like I said, we don't know for sure, but we know that this very, very wasteful process takes place in C3 plants when it's too hot and we don't have enough carbon dioxide and we have way too much oxygen. But some plant species have alternate modes of carbon fixation to avoid this. So some species have evolved to avoid this. I'm just going to make this really big. I'll be right back. There we go. Some species have evolved to avoid this. We have alternative modes of carbon fixation to minimize photorespiration. Alternative modes of carbon fixation to minimize photorespiration. This usually happens in hot, arid climates. Climates where they have to deal with low CO2 levels and increased transpiration if they don't close their stomata. So plants that have to adapt to kind of overcome this process because it would be not so great and cost a lot of energy to kind of keep up with photorespiration if they didn't have some way around it. So there are two main types of alternative carbon fixers. We're going to call them C4 plants and CAM plants. I said we're going to call them. It's what science calls them. There are C4 plants and CAM plants. So they have a different way of kind of going about this process. Goodness gracious. Yes, okay. We're going to start with C4 plants. Some examples of C4 plants, corn and sugarcane. On really, 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 really hot, dry July days, corn can go through it, right? Like they have to kind of figure out another way to fix carbon dioxide without losing all of the water that would have gone out of the stomata on those really, really hot, dry days. What they do is they preface the Calvin cycle with an alternate mode of carbon fixation that forms a 4-carbon compound as its first product. So we have a preface to the Calvin cycle. So right before the Calvin cycle, what feeds into the Calvin cycle, we have an alternate form of carbon fixation resulting in a 4-carbon compound as its first product. And that is why we call it a C4 plant, because the first product is a 4-carbon sugar. This process of prefacing the Calvin cycle and creating a 4-carbon compound first is believed to have evolved independently at least 45 separate times. Believed to have evolved independently at least 45 separate times. Meaning on their own, without a sharing of genetic information, this process has popped up 45 times. Which is insane. Like, they did this without communicating with one another. They just figured out that they could use stuff that they had to minimize the loss of carbon dioxide. And this is how they did it. Let's see. Anatomy is really, really important to this. Or cell anatomy. Meaning they have to have these cellular structures to be able to carry out this process. And the specific anatomy is 2 types of cells. They have 2 types of cells that help them carry out this process. They are mesophyll cells and bundle sheath cells. So if you remember from our very, very, very first photosynthesis, like anatomy of a plant, mesophyll cells is that like the guts of a leaf, right? So if we've got our leaf, this is our leaf. We've got our like outer layer of the leaf with our more protective cells and our mesophyll in here. And that's where the bulk of everything happens. So these cells have these mesophyll cells, but they also have these bundle sheath cells. Bundle sheath cells are arranged into tightly packed veins. So if you look at a leaf and it's got those like really tough veins running through it, those are the bundle sheath cells. So let's see, bundle sheath cells. Are arranged in tightly packed veins. So our Calvin cycle is confined to the chloroplasts of bundle sheath cells. So this is where our Calvin cycle takes place. Calvin cycle confined to the chloroplasts of these cells. So in C4 plants, the mesophyll cells are only for the light reactions, and the bundle sheath cells are where the Calvin cycle takes place. But here's our catch. The Calvin cycle is preceded, like we said, by the incorporation of CO2 into organic compounds in the mesophyll cells. It's preceded by the incorporation of CO2 into organic compounds in the mesophyll cells. So we've got our mesophyll cells. They will carry out the light reactions just like normal. And then they will do this like little mini preceded cycle where they'll incorporate CO2, but they'll put it into organic compounds. Then they will send those organic compounds to the bundle sheath cells, and the bundle sheath cells will take those organic compounds, go through the Calvin cycle, and give us our sugars at the end products. So this happens in 3 steps. Step 1, our enzyme in a mesophyll cell called PEP carboxylase adds CO2 to a compound called PEP, which gives us oxaloacetate, which is something that you guys, I think it was in the Krebs cycle, but that's not necessarily super important to this cause. So enzyme in the mesophyll cells will add carbon dioxide to PEP. So the enzyme in the mesophyll cells is called PEP carboxylase. Yeah. We'll add CO2 to something called PEP. And that will give us oxaloacetate. This happens because PEP carboxylase can fix carbon when Rubisco can't. So Rubisco's great. We love Rubisco. Like I said, one of the most common proteins on Earth. But it's not perfect. So whenever our CO2 levels are too low, or if the situation is just not right, instead of Rubisco fixing carbon dioxide, PEP carboxylase will do it. This will give us our 4-carbon product, which is that oxaloacetate. Our 4-carbon product is exported to the bundle sheath cells. If you remember our cell junctions conversation, that happens through junctions called plasmodesmata. So this allows the stomata in the mesophyll cells to stay closed. So if our mesophyll cells don't have our stomatas open, that means we can't lose water from where the light reactions are taking place, which is what needs the water in the first place. So by closing all the mesophyll stomata and only opening the stomata in the bundle sheath cells, we've minimized the amount of water that can leave the cell. So we've got our 4-carbon product happening in the bundle sheath cells. It is exported to the bundle sheath cells through plasmodesmata. That doesn't seem right. Oh well, okay. Step 3. Our 4-carbon sugar will release CO2 in bundle sheath cells. That is fixed by rubisco. And that happens, like we know, in the Calvin cycle. And that will generate pyruvate. Yep. So, and that will get sent back to the mesophyll cells. And those mesophyll cells will use ATP to turn pyruvate into PEP. That seems a little bit complicated, and I might have gotten confused in there a little bit. So, our Calvin cycle happens in our bundle sheath cells. But our bundle sheath cells aren't open to the environment. That's where I was confused. So, the stomata are more so open in the mesophyll cells. The mesophyll cells will go through the light reactions and will go through this kind of fixation of carbon by PEP carboxylase and send their products to the bundle sheath cells. The bundle sheath cells will use that product, go through the Calvin cycle like normal, and produce pyruvate. Pyruvate will get sent back to the mesophyll cells, where they will use ATP to turn that pyruvate into PEP, which will go back into that PEP carboxylase kind of cycle. So we've essentially just added a bunch more cycles to it. This little kind of idea, there is no linear electron flow. Only cyclic. This separation of the two cycles, the separation of the light-dependent reactions in the Calvin cycle, means that linear electron flow isn't going to work as smoothly. So they just don't do it. They only do this cyclic electron flow, which, if you remember, doesn't produce as much ATP but still gets the job done. So that is our C3 plants. I promise CAM plants are a lot simpler. So finally, we've got CAM plants. That should be in all caps. So our C4 plants were like grasses, corn, sugarcane. Our CAM plants are like cacti and pineapple trees. The way CAM plants do it is they open their stomata at night. So in the desert, these plants have adapted to where they only open their stomata to suck in carbon dioxide to let out oxygen at night, because if they were to open it during the day, that really, really hot sun would suck all the water out of them. The main goal here is to conserve water. So while the stomata are open, they do everything they can. They take in CO2, And when they take in CO2, they make organic acids. Because we have to have a way to store this carbon dioxide until the light reactions start up the next day, because they can't work independently of one another. The Calvin cycle relies on the light dependent reactions for those electron carriers, right? So they can't happen independently of one another. They interact with one another. So we have to have a way to have carbon dioxide for the Calvin cycle to work during the day when the light reactions are going. They do this by opening their stomata at night, using CO2 and making organic acids. This process is called crassulation. CRASSULACEAN, acid metabolism, or CAM. So our C4 plants had a 4-carbon first product. Our C3 plants had a 3-carbon product. Our CAM plants make organic acids. Our mesophyll cells store these organic acids in vacuoles. Until morning. In the morning, those stomata closed. One more. So during the day, we have our light reactions. We're just going to write can. Sorry. Our light reactions can supply NADPH and ATP. For the Calvin cycle, and those and CO2, is released from organic acids. No, it's not the most riveting thing for your Monday morning, but it's kind of cool to learn about the different ways evolution has come up with kind of working its way around this photorespiration thing. Let's see. Don't have a heck of a lot of time left, but I do have a little diagram. I'll give you time tomorrow to fill out the exit ticket because there's not a heck of a lot of time left. But I wanted to show you this. So we're just gonna have these little colored boxes because I think it helps. It's not the best, but it will do. So if we're comparing and contrasting our C4 and our CAM plants, our C4 plants are divided up by mesophyll cells, and our bundle sheath cells. And our CAM plants are divided into night and day. But we have our CO2 coming in to our mini cycle where we have organic acids pen's really big. It says organic acids, which send CO2 into the Calvin cycle, which releases sugar. And the exact same thing happens with CAM plants, where we have CO2 coming in, we're storing our organic acids, which are released as CO2 into the Calvin cycle. Which give us our sugars. So it's a very similar process. They just go about it differently. One of them has different cell differentiation, and the other has what's the word? A kind of temporal dislocation of the two. So I know there's a lot. Does anybody have any questions? All right, I will give you time to do the exit ticket tomorrow. If you think of anything, let me know. And then we will start on our pre-lab then.